There we go. All right, we started recording and I'm going to get started because I like to start on time. Come on. Come on. Don't do this to me. Every time I use Zoom, the computer complains. Okay, now what's this? Screen one. Okay, no. Okay. Screen. Stop it. 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 Okay, let's try this again. Share. Gosh, sometimes I really hate Zoom. Oh, there it goes again. It's okay. All right, there it goes. Okay, it's time to start, so I'm starting. I start on time. Nobody's here, I'll talk to an empty room. So we were talking about how enzymes speed the rate of reactions. Now, in order to speed the rate of reactions, they're not going to affect basic parameters of a reaction. To explain what I mean by that, let's use I'm going to use our makeshift blackboard here, which is our Zoom blackboard. And all right, now you can see it. So if you have a reaction, let's say this reaction goes something like this. You have A plus A and it goes to, let's say, HA, something like that. So in chemistry, you have this thing called the equilibrium constant of that reaction, and that equals the concentration of the products divided by the product of the concentration of the reactants. All right, enzymes don't change this. This doesn't change. That's inherent to the reaction. And then there's something called the Gibbs free energy of a reaction. And the Gibbs free energy is related to this equilibrium constant. It's related by an equation that you don't have to know, but it's a gas constant times the temperature, and that temperature is in Kelvin. And it's times the natural log of the reaction constant of this. And because this doesn't get changed, and because these are constant, because that doesn't change, enzymes don't change this either. So enzymes don't change inherent properties of the reaction. What enzymes change is what's called in books, they call it, they do this. And what does this mean? This means that's a symbol that's put in to confuse people, basically. This is the energy of the intermediate. And if you draw a graph of just energy versus the progress of the reaction through time, you have the energy of the reactants, the energy of the products, and there's some sort of energetic hump the reaction has to go through. So the difference between this transition point and here, that's the so-called G sub double star. And I don't know why they do that, but they just do. Why not call it TS, transition state? I don't know. They call it what they call it. And I know it confuses the daylights out of people, but that's what this is. And that's what enzymes change. So enzymes change this. That's what they change. So you have a delta G for reactions. And if that delta G is positive, that means a reaction is not spontaneous. Meaning that it's thermodynamically not favored. For this type of reaction to go, you'll have to put a lot of energy into it. Some reactions have a delta G of reaction that's negative, and these are spontaneous. And so these are thermodynamically favored. Now, you might think, well, if it's positive, it should be favored, right? Well, if it's positive, what that means, we go back to our graph here, it means that the energy of the products is higher than the energy of the reactants. So the reaction would have to essentially go uphill. That's what that means. Whereas if the energy of the products is lower than the energy of the reactants, that means that the delta G of that reaction is negative. So these are thermodynamically favored, spontaneous. These are thermodynamically not favored, not spontaneous, if the delta G value is positive, or in this case, if the delta G value is negative. So let's go from our makeshift blackboard back to thermodynamic graph of a spontaneous reaction would look exactly like that. And you have this energetic hump to get over, that's your hurdle, and enzymes give the hurdle a haircut. That's how they work. There it goes again. Why it does that?
It's over on the,
Sorry?
it's over on the slide you can go to.
See, I can't see that here. Let me do this. Project. No, I don't want to, I don't know why it keeps changing that. Okay. Okay, you. There. All right. All right, so the laws of thermodynamics are not changed by enzymes. That's the point of all this. And if for a reaction to go, you have to put energy into it. Doesn't matter if it's enzyme catalyzed. So how much energy does it need? What's the inherent energy of the enzyme? Gibbs free energy describes that. That is this number, delta G. That's a Gibbs free energy. And the Gibbs free energy typically is the sum of the changes in the heat energy known as enthalpy and the change of the, that's in order, or entropy. And so the enthalpic change is something called delta H times the temperature. And then that is summed with the change in entropy, or delta S. And so this is the point I was making earlier, that if mathematically, if delta G is positive, excuse me, if it's negative, the reaction spontaneous, it will proceed in the forward direction. You shouldn't have put energy into it. At least get the reaction. You do, you get the reaction to be initiated, but you don't because you're going the forward direction. If, however, the delta G is positive, then it's not thermodynamically spontaneous. You have to put energy into it. There are a lot of reactions in your body that are not thermodynamically spontaneous. So what does your body do? It takes thermodynamically favorable reactions and couples them with reactions that are not favorable. This is called reaction coupling. Very, very common strategy in your body. So let's take, for example, the conversion of something, we're going to call it A, to something else, we're going to call it B. That's unfavorable. How do you get it to go? You take another reaction, namely the conversion of C to D that is favorable, and you put them together. So now the reaction of A plus B to C plus D, that's favorable. Think of A plus B. Think of A to B as your car, going from point A to point B without any gasoline. You have to put energy in it. You have to push it. So C to D, let's say that's the movement of a tow truck from the same place to another place. Now we're going to take your car without gas, and we're going to hook it to the tow truck, and that will now get the car to the place you want, as the tow truck is going there too. You've coupled the movement of the car with the movement of the tow truck. You've coupled one reaction that's unfavorable to another reaction that is favorable. That's how your body does business. So your book uses these cogs as an example. That's, I think, a pretty good way to think about it. You have reactants going to products. That's your favorable reactions, your so-called exergonic reactions. You couple them to unfavorable reactions, endergonic reactions. Notice as the one wheel moves, its cogs will drive the movement of the other wheel, the less favorable reaction wheel. And that's how reaction coupling works. So the reaction that's used in cells is the hydrolysis of ATP. You take ATP, you add water across the phosphoanhydrate bonds, you get ADP, adenosine diphosphate plus inorganic phosphate. That reaction is favorable. By coupling that with other reactions that are less favorable, you drive those less favorable reactions. So here is ATP. We use our pen function here. You look at this bond right here. That's called a phosphoanhydride bond. Notice that you have these two oxygens that are negatively charged right next to each other. They're going to tend to repel each other. Because they're going to tend to repel each other, if you stick a water across this bond, boy, drawing using the mouse is really hard to do. If you stick a water across that bond, you break that bond. You get these two particular negatively charged atoms away from each other. That reduces tension. This is subject to resonance stabilization, as is this. Therefore, your end products have a lower energy than the reactant itself. And this favorable reaction, therefore, this product is very high energy. The products have much lower energy. That is a very favorable reaction. By coupling this hydrolysis of ATP to ADP plus inorganic phosphate, you drive a lot of unfavorable reactions in cells. So how do you make ATP in cells? You use reactions that are called catabolic reactions. Catabolic reactions. Catabolic reactions will take large molecules, break them down to smaller molecules, and they will generate ATP, and they will generate reductant. Reductant means you're taking high energy electrons and loading them onto electron carriers. Anabolic reactions take small precursors and make larger molecules. So anabolic reactions build things, but those are energy demanding. You will expend ATP in order to perform anabolic reactions. So to generate ATP, one of the first energy yielding reactions is glycolysis. And glycolysis will break down monosaccharide sugars, in particular glucose. Now, glycolysis does not require oxygen. So all organisms, be they aerobic or anaerobic, can use glycolysis. You generate ATP during glycolysis, but all the ATP you generate is by means of substrate level phosphorylation. Substrate level phosphorylation generates ATP by making a phosphorylated intermediate, and that high energy phosphate bond is transferred to ADP to make ATP. That's substrate level phosphorylation. You don't make much ATP with substrate level phosphorylation. You make enough to survive. You don't grow very fast, though. Now, if an organism generates all of its ATP via substrate level phosphorylation, then that organism carries out a fermentation. And its metabolism is so-called fermentative. So I realize that your idea of fermentation is probably organisms that take glucose and they convert it to ethanol, right? That's probably what you think of when you think of fermentation. Is that fair? Well, it turns out this is one type of fermentation. One type of fermentation. Because there are lots of ways to take glucose and break it down. Some organisms can take glucose and they can break it down, but instead of ethanol, they'll make a 4-carbon acid called butyric acid. That's also fermentation. There are other organisms, for example, organisms involved in the making of Swiss cheese. They take glucose and they ferment it to propionic acid. That gives Swiss cheese its sharp taste, is a propionate. That's also fermentation. So when you hear fermentation, don't just think ethanol. Let me give you an example of how this got a very high-class lab in Boston in big trouble. So when I was in graduate school, there's a disease of the central nervous system. It's called cryptococcosis. You get this from lots of different sources, but one of the biggest sources of this is pigeon dung. Pigeon dung is aerosolized, the organism which normally grows as a filamentous fungus in the soil. when you inhale it, now it becomes a yeast. So it buds like a yeast. So because it forms a yeast, a high-priced outfit in Boston said, oh, if we can detect ethanol in the blood, that's a diagnostic test for cryptococcosis. We should be able to detect it in the cerebrospinal fluid. If we can, that means you have cryptococcosis. So my PI and I, we read this paper, we went, I don't think so. So we took the organism, it's called Cryptococcus neoformans. We grew it in a special culture that would make it grow as a yeast. And then we took this and then we put it in a gas chromatograph. And we discovered you got lots of different things and a little tiny bit of ethanol. So little that if you tried to detect it in cerebrospinal fluid, you wouldn't be able to detect it. Oh, what if you had a really heavy infection? Well, then maybe you could detect it, but by then your patient would be dead. So we published this article and the high-class outfit in Boston got really mad at us. And they got deceived by essentially getting this idea that the word fermentation automatically means ethanol. In fact, I was at a church once and somebody said, well, in the Garden of Eden, there couldn't have been yeast because there was no fermentation. What's wrong with that argument? Fermentation came about as a result of the fall because nobody got drunk until after the fall. No one got drunk after the fall. That's the first time in recorded history of people getting drunk. I wanted to raise my hand and go, excuse me. Tried to talk to him afterwards. He didn't want to talk to me. I just got one of these personalities. I just put people off, you know. His mistake is that he's thinking fermentation equals ethanol. There are lots of soil organisms that are degrading leaf litter that are fermenting it. Otherwise, Eden would have been overrun with dead leaves. So, yeah. So if you make your ATP solely by means of, wait, I thought I closed you. There. So if you make your ATP, if you're an organism, you make your ATP through substrate level phosphorylation, you carry out a fermentation and you will make some sort of reduced organic process, reduced organic compound. Well, how do organisms like us, how do we make the lion's share of our ATP? We make it via a process called oxidative phosphorylation. And in this case, you're going to use proton gradients set up across membranes. And you're going to readmit those hydrogen ions, and you'll use a ATP synthase to make ATP. This requires membranes. Substrate level phosphorylation does not require membranes. Oxidative phosphorylations do require membranes. Incidentally, our electron acceptor is oxygen. That's why we have to breathe in oxygen. Anaerobic organisms can also carry out oxidative phosphorylation. They just use something other than oxygen. They use things like nitrate or sometimes nitrite. In fact, denitrification, which converts nitrate to nitrogen gas, that's a result of this process. You can use sulfate. You can use other types of electron acceptors as well. You don't have to use oxygen. Oxygen is the best one. But if you get stuck in a pinch, you can use something other than oxygen. So let's take a look at glycolysis again. Remember, I told you have to know what goes in and what comes out. So what goes in is glucose. Glucose. Can fructose go in? Yes, but fructose is ultimately converted to glucose. Can galactose go in? Yes, but galactose gets converted to glucose. So all roads lead through Rome. All roads lead through glucose when it comes to glycolysis. So glycolysis is converted to 2 3-carbon compounds. How many carbons are there in glucose? 6. C6, H12O6. 6 carbons, you convert it to 2 3-carbon compounds called pyruvate or pyruvic acid. Pyruvate gets transported into the mitochondrion, where it will undergo further oxidation. That's not part of glycolysis. So glycolysis is this process here. You don't have to know the steps, don't have to know the intermediates. You need to know this, these, and everything that is circled in color. That's what you need to know. So you burn 2 ATP at the beginning to prime the pump. You will generate 2 reduced NADs, so 2 NADHs. You have 2 sets of molecules with reducing equivalents, and then you'll make 4 ATP, but if we calculate net ATP, it's 4 minus 2, so your net output is 2 ATP. So what goes in? Glucose. What comes out? 2 NADHs, 2 ATP, and 2 pyruvates or pyruvic acids. That's what comes out. So let's rehearse this again. Emma, what goes into glycolysis? No, it goes in.
Oh, glucose.
Glucose. And then let's go, what comes out? We'll just go through here. Name one thing comes out. 2 ATP, what else? And 2 pyruvic acids. That's glycolysis. That's all you need to know about glycolysis. Pyruvate gets sent to the mitochondria. In the mitochondria, it's converted to acetate. And that acetate is linked to a carrier called coenzyme A. Coenzyme A is going to carry acyl groups. Coenzyme A is made using the vitamin pantothenic acid. Pantothenic acid. Coenzyme A, by the way, the acetate, in that process, you generate 1 more NADH. So I'm going to go back to our little blackboard here. So inside the mitochondrion, you have the outer and the inner mitochondrial membrane. MM is not M&Ms. That's not the candy. That's mitochondrial membrane. This is the inner mitochondrial membrane. In here, this is called the matrix. Obviously named after the movie, right? Oh, come on. You guys have heard of the Matrix, right? Please. Thank you. Good. I'm not that old. Well, actually I am, but. Now in the matrix, you have lots of soluble enzymes. One of the enzyme complexes in there is called the pyruvate dehydrogenase complex. My word, that's a mouthful. So this is the PDH complex. big, 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 big enzyme complex. That's in the matrix. And this is going to take your pyruvate that's pyruvate. You don't need to know the structure. I'm just drawing it out to show you what happens to this molecule. You will get from this a carbon dioxide, plus you get a reduced, and then you're going to have a coenzyme A that's going to come in here, and you're going to have there's your acetate, and it's linked to coenzyme A like that. So this is called acetyl coenzyme A. That's what's going to go to the next set of enzymatic reactions. So you're going to generate more reductant, you lose carbon here, and now you set yourself up for the next set of reactions. There we go. All right. In the mitochondria, the acetyl-CoA will enter a cyclic set of reactions called the Krebs cycle. Krebs cycle enzymes are also soluble in the matrix, with the exception of one that's embedded in the membrane. That's succinate dehydrogenase, but you don't need to know that. And this will completely oxidize the acetate to carbon dioxide and water, and you'll liberate lots of high energy electrons. So what goes into the Krebs cycle? An acetyl coenzyme A. You will generate 2 carbon dioxides, 3 NADHs, 1 reduced flavine, 1 FADH2, and a GTP that you make via substrate level phosphorylation. So what goes in? 1 acetyl coenzyme A. What comes out? 2 CO2s, which makes sense. You have 2 carbons in the acetate. 2 carbons, so they come out as 2 CO2s, 3 NADHs, 1 FADH2, and 1 GTP. Now, how many acetyl coenzyme A's are going to be generated per glucose? 2. So how many ATPs are you making per glucose? You take all that and you double it. So for per glucose, you make 6 NADHs, 2 FADH2s, 2 GTPs, and you'll make 4 carbon dioxide molecules. And here's the Krebs cycle. I like this summary better because it essentially shows you generate a 6 carbon compound, you come out with a 4 carbon compound, 2 of those CO2s come off at 2 of those carbons come off of CO2, and you'll generate reductant as well. So here's your PDH, complex catalyzed conversion of pyruvate to acetyl coenzyme A. Goes through the Krebs cycle, and there you are. So per sugar, we add it all together. We have 6 from the Krebs cycle. We get 2 from the PDH catalyzed complex, 2 from glycolysis. That gives us 10 molecules of NADH, 2 FADH2s, and 4 molecules of ATP because we have 2 GTPs, 2 ATPs from glycolysis. A GTP energetically is the same as an ATP, and that's what we have. So we're doing all that and we're generating ATP. And we're also generating NADH, and we're producing carbon dioxide and water. Now, you have a problem as an organism. You want to know what that problem is? See these reduced carriers? You can't oxidize any more glucose until you unload those high energy electrons. Otherwise, you don't have any carriers to accept high energy electrons from any more glucose. So you need to get rid of those. What do you do with them? You give them to an electron transport chain. And the electron transport chain will carry those electrons down the chain, and it'll carry it to molecular oxygen. And it will reduce molecular oxygen to water. And in doing so, you convert something that is potentially deleterious to cells into something that's completely innocuous, water. Now, there's something else that happens on the way to the store, and it's this. The electrons are given here thermodynamically. This transfer of electrons down the chain is very spontaneous. So this reaction from here all the way down to here, this has a very negative delta G. Since this has a negative delta G, I wonder if you can use that to do work. The answer is you can. So you're going to alternate in this carrier between carriers that carry hydrogen ions and electrons and carriers that just carry electrons, like cytochromes. Those hydrogen ions will be extruded on the other side of the membrane. So as these carry their electrons, the extra hydrogen ions, they get pumped essentially to this compartment between the outer mitochondrial membrane and the inner mitochondrial membrane. And as you take electrons down this chain to molecular oxygen to make water, you're going to keep pumping hydrogen ions into this intermembrane space. You are now going to create a pH gradient between this space and this space. So the pH here, relatively speaking, is going to be relatively high. With all these hydrogen ions out here, the pH out here I wish I had a magic pen that would do this, but I don't. pH out here is, relatively speaking, is going to be low. So now if you readmit these hydrogen ions back into the matrix, that's going to release a lot of energy. You can use that to do work. So here's the energy transducing molecule. This is the ATP synthase. The ATP synthase. And the ATP synthase brings these protons back into the matrix, but in doing so, it takes ADP and inorganic phosphate and converts it to ATP. And that is the essence of oxidative phosphorylation. You use membranes, you transfer electrons down that electron transport chain, which is embedded in the membrane. You create a gradient of charge and of pH and of concentration between the two compartments. That gives you the potential to do work. And then you readmit those hydrogen ions using an ATP synthase to do exactly that and to make all the ATP you need. And this generates bucket loads of ATP. bucket loads. So you make about 2.5 ATP per NADH, and you make about 1.5 ATP per reduced flaving. And with 10 NADHs, that's 15. Sorry, that's 25 ATP you make right there. You have 2 reduced flavings, you make 1.5, that's 3. So that's 25 plus 3, that's 28. You then make 4 more, that's 32. To make 32 ATP per glucose. That's impressive. That's impressive. That's a lot more than the 2 that you made for glycolysis, isn't it? So this is a picture just ripped off your book, and you can see the electron transport chain comes in complexes, complex 1, 2, and 3, and 4. So electrons go from 1 to 3 to 4, or they go from 2 to 3 to 4. Cytochrome C carries electrons between complex 3 and complex 4. That's why cytochrome C is so important. These are going to pump protons, pump hydrogen ions into the intermembrane space. And then this is your ATP synthase that will make ATP by readmitting these hydrogen ions back into the matrix. And I have good gravy. Okay, let's watch this video here. Your session has timed out. You need to log back in. What? Oh, you need to log back in here. So, there, okay. Right, let's go ahead and bring this up.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron, a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme, and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
So this is my mnemonic. I don't know if this will help you. Oil rig. Oxidation involves loss of electrons. Reduction involves gain of electrons. Oil rig. I don't know if that helps you. If it does, great. If it doesn't, well, then think of something else. All right, let's now take a look at, oh, sure. Man, that is annoying. I find mnemonics helpful. My nursing students definitely find mnemonics helpful. So they roll their eyes at a lot of my mnemonics, but I still think they help them, even if they do roll their eyes at them. You'll roll your eyes at plenty of mine, don't worry. They get pretty bad sometimes. Now, I'd like us to consider this one that's going to show you how oxidative phosphorylation works.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. The terminal electron acceptor and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Enough of you. And this table, this shows you, this is from your book, this shows you precisely, this is the best stoichiometry available so far. Your high school textbooks, I still see 38 in high school textbooks. That number has been known to be wrong for the last 20 years. So I don't know why high school textbooks are still reporting 38, but the number is 30, 32, in that range, essentially. And this essentially is where we are, but you can see this gives you a lot more ATP than does substrate level phosphorylation. So that's how you break down glucose. What about fatty acids? Fatty acids are admitted into the mitochondrion and they're linked to coenzyme A. And then they're broken down 2 carbons at a time into acetyl coenzyme A. That process is called beta oxidation. Beta oxidation. And then acetyl coenzyme A goes through the Krebs cycle. And your book shows you essentially how this occurs. These are the steps of beta oxidation. You don't need to know those. The only thing you do need to know is that you yield from this some reduced flavine, some NADH. This then produces acetyl coenzyme A that goes straight to the Krebs cycle. What if you have an odd numbered carbon fatty acid? Then your last component is propraneal coenzyme A. Propraneal coenzyme A is converted into succinyl coenzyme A. That still enters the Krebs cycle. So regardless of the number of carbons in your fatty acid, it all goes through the Krebs cycle. And acetyl coenzyme A is used to make several things. It's used to make cholesterol. Cholesterol really is only a polymer of coenzyme A. Cholesterol is then used to make steroid hormones and biosalts. Acetyl coenzyme A is used to make ketone bodies when your body goes into ketosis. And it's also used to make fatty acids that are used to make either triglycerides for storage, fats for storage, or phospholipids. So coenzyme A is a really good nexus between catabolism and anabolism. What about amino acids? You break down proteins to amino acids. How are those catabolized? Amino acids are shunted either into glycolysis or they're shunted into the Krebs cycle, one or the other. Now, what happens if your body needs to make glucose? Glucose levels start to drop. Let's say practice went long, the DC was closing, you get home, you really don't feel like cooking. You look in the fridge, oh man, my roommates ate that chicken that I had from the other day. Oh, it was probably old, he, jokes on them. They'll probably have sour stomachs. But you say, oh, I don't know, I need to study, so maybe you grab an apple or something like that. And then middle of the night, you wake up and your stomach starts growling and you say, this is ridiculous, I can't eat, it's the middle of the night, I need to sleep. So what's happening at that time is your body's glucose levels are dropping. Glucagon levels become high, and now it tells your liver to make glucose. And that process is called gluconeogenesis. So the body takes small molecules and it puts them into the glycolysis pathway, and it runs glycolysis in reverse. So all the reactions of glycolysis are reversible except for 3 of them. The conversion of pyruvate to phosphatidylpyruvate, that's not reversible. The conversion of fructose 1,6-bisphosphate into you can those reactions are reversible, but you have to use a different pathway. You have to use a different set of enzymes. So this particular conversion requires you to make succinic acid first before carbon acid. Then you convert that into phosphatidylpyruvate. Phosphatidylpyruvate is then shunted through glycolysis, gluconeogenesis. So this pathway is reversible, and that's called gluconeogenesis. Some amino acids can be used to make glucose, others can't. If they can be used to make glucose, they're called glucogenic. Others, however, if they can't be used to make glucose, you can still use them to make ketone bodies, and they're called ketogenic amino acids. You cannot take fats and use them to make glucose. Plants can do that. We can't. We don't have enough reductant to do that. Plants have a bypass pathway that lets them do that, called the glyoxylate pathway. We don't have the glyoxylate pathway. So we can't make sugar from fats. Sorry. That's why a diabetic, you can't give a diabetic whipping cream, essentially, to raise their blood glucose level. It won't work. They can't take fat and use that to make glucose. They have to have glucose. They have to have honey, a sugar cube, that disgusting goo that they'll carry with them, that sort of thing. Sugar tablets, you can buy glucose tablets, usually for free sometimes at the drugstore. You just take one of those, suck on them, put them under your tongue. Usually that raises the blood glucose level. So that's how you break things. How do you make things? How do you make things? As we said, we're not quite there yet. Some amino acids, they enter the Krebs cycle, others enter glycolysis. The upshot is that you will always be able to use them to make glucose. Now, if you want to make things, say you want to make a polysaccharide, you just can't link sugars together. That's an energy dependent process. You have to activate the sugars. So what you do to activate them is you take uridine triphosphate, UTP, which is made from ATP. You're going to take the UTP and link it to the sugar, and you're going to make the UDP sugar, UDP glucose, UDP fructose, UDP galactose, whatever. You use those to link the sugar together. And the polysaccharide synthase will have a binding site for the UDP site. By activating the sugar, you prevent the sugar from leaving the cell. For lipid synthesis, you'll use acetyl coenzyme A, and you'll carboxylate it to malonyl coenzyme A. That enzyme, acetyl coenzyme A carboxylase, is one of the major enzymes that's part of the control nexus between whether you make fat or you don't make fat. So you'll use acetyl coenzyme A to make fatty acids, then they're linked to a glycerol bridge, and that's how you make fats and phospholipids and things like that. Amino acids, we can make 8 or 9 of them. Those are made, well, we can make about 12 of them actually. Those are made usually from precursors that are made either from glycolysis or the Krebs cycle. So glucose 6-phosphate is used to make histidine. 3-phosphoglycerate, that's used to make serine. Serine is used to make cysteine and glycine. And then Krebs cycle intermediates. So alpha-ketoglutarate, which is an intermediate from the Krebs cycle, that's used to make glutamate and glutamine and arginine and proline. And this shows you here essentially how you do that. And then oxaloacetate is used to make aspartate. And then aspartate is used to make these other amino acids as well. So this is used to make oxaloacetate, used to make aspartate and asparagine as well. Succinyl coenzyme A, which is not on here, that's used to make heme groups and what have you. So the Krebs cycle also makes precursors for biosynthesis. So it is a so-called amphibolic pathway, A-M-P-H-I-B-O-L-I-C, amphibolic pathway, because it's used to break things down, but it also makes precursors to make things. If you eat a lot of protein, those amino acids have to have the amino groups removed, and then the carbon skeleton is dealt with, but that ammonium is really toxic to cells. So what does your liver do? It takes the liver and it puts them in something called the urea cycle. And it makes urea, and urea is the main nitrogenous product that is in urine. This is the reason why, if your liver goes south, you're subject to something called ammonification. You get ammonium in your blood, and that's really toxic to your blood. It interferes with neurotransmitter synthesis in the brain, and it raises the pH of the blood, and it's very, very dangerous. That's why liver damage is so bad for you, because the liver takes ammonium and makes urea, and urea is not toxic. And so here's a list of the essential amino acids that you need in your diet. Here's the ones that you can make. And you make these from either glycolytic or Krebs cycle intermediates. These we have to have in the diet. 1, 2, 3, 4, 5, 6, 7, 8, 9, as you can see here. Some of these too, under certain conditions, you will need in the diet if you are, say you're working hard, say you're lifting weights, you'll actually need more arginine in the diet, say you're lifting weights. Children need histidine. Adults don't need much histidine, and so on. And so some of these become essential under certain conditions and what have you. Very good. That is the end of today's lecture. So I'm still on jury duty for this week. I didn't get the call. My number didn't come up for today. May come up tomorrow, may come up Wednesday. I don't know. I'll keep you in the loop. I'll let you know if I'm not here Wednesday, I'll do some sort of video, something later on in the week. So look for that. And everybody, please wish Griffin a speedy recovery. So everybody wave to Griffin and wish him a speedy recovery. Yep, Griffin is waving violently there. So he got the flu, so that's why he's out for, hopefully, Gilbert. Griffin, you feeling all right? No, hopefully you're on the mend. So we hope to see you soon. So, all right, so that's it for today. And we will see you guys later. Okay. And this.